Good afternoon. I'm Arthur Popper, and my colleague Tony Hawkins and I are very delighted to be able to present the keynote talk to you today. Tony is the gentleman in the picture with the hair, and he will be presenting a few of the slides that come on in the talk. This talk is going to consider the role of sound in the lives of fishes. We're also going to discuss a topic that Tony and I have been working on for the last 15 or so years, the effect of man-made or anthropogenic sound on fish. We're going to some degree, focus on gadded fishes. These are the cods and relatives, primarily because the Atlantic cod is really one of the most important fishes in the world commercially. There's also a good deal of data on bioacoustics of Atlantic cod and other related species. And in a couple of papers that we've been publishing in JASA recently, we make the point that cod could actually serve as a model species for understanding hearing and also effects of noise on aquatic life. As a way of outline, we're going to start out and talk a tad about how fish hearing evolved, how vertebrate hearing evolved. We'll then talk about the nature of underwater sound as a bit of a primer. We're going to talk about sound production and acoustic communication by fishes, focusing on gaddids. We'll talk about sound detection mechanisms, and we're going to digress here to talk about some ideas we have about how to determine fish hearing and what hearing really is. We'll then go on to discuss potential effects of anthropogenic sound on fishes. And here we're going to present a bit of data from two studies that we've done, one by myself and one by Tony. And then we'll talk about setting of guidelines and criteria, something that we've been engaged in very heavily over the last several years. Let's start out considering, why did hearing evolve? Well, if you think about the most primitive fishes several hundred million years ago, the environment was probably living in murky water. The ability to detect signals around them that are of biological relevance was probably limited to the distance of touch or vision or whatever senses they had. But they really did not have any sense of the greater world around them. So Dick Fay and I proposed a number of years ago that the evolution of hearing would provide fishes with a much greater percept of the world, much greater area over which to detect signals. It basically enabled fishes, no matter what the environment was like, to detect predators and prey and to learn about their acoustic environment. So the fishes basically evolved hearing, not for communication, but for detection and use of what we call the acoustic scene. In effect, it allows fishes to be able to know the world around them at some distance from them. And ultimately, communication probably evolved, but that was not the primary source of sound detection. But a very important point to make that's relevant to the whole talk is that anything that interferes with sound detection and the use of the acoustic environment can affect the fitness and survival of fishes. They require this sound, virtually all species do, and so interference with detection has a potential impact on the animal. Just briefly, let's review underwater sound for those of you who don't think about this very often. If you want to get more, we recommend a website, delegates.com, where they cover underwater acoustics in a very, very effective way. In air, we primarily think of sound pressure because particle motion, the second component of the sound, doesn't travel very far due to the low density of the air. However, in water, particle motion is very substantial, and because of the density of the water, and so at substantial distance from the source, you still can have both pressure and particle motion. Significantly, fishes and invertebrate are primarily sensitive to particle motion. Pressure detection is a secondary evolution issue, and not all fishes use sound pressure. Just think of sound pressure and particle motion as this sound here, starting from your left. The sound waves, which are sound pressure, travel at a distance from the source. They are a scalar quantity. They do not have direction. At the same time, the particles in the water move back and forth, staying in about the same spot, but because they move back and forth in the direction of the sound source, they actually provide an opportunity to provide directional information to the animal. So it's very important that particle motion is the major component of hearing for fishes and invertebrates, and it can provide information about sound source direction. While we've always thought about particle motion and sound pressure when it comes to fishes, a new idea has recently started to perk up, and that is the fact that in water, there's also something we call substrate vibration. Not many people have considered this, particularly with regard to fishes, but it's something that we think that needs to be considered in the future, particularly when we're talking about things like pile driving or seismic air guns, where sound is projected into the substrate. As a consequence, the sound travels through the substrate. It can travel very fast, and ultimately it comes out of the substrate. And this can have an impact on animals at a substantial distance from the source. It also means that animals in the substrate, living on the substrate or just above the substrate, are not only getting direct stimulation from the source through the water, but also getting stimulation from the substrate itself. And this can potentially be very significant. But as I say, we know very little about it. So many fishes use sound for behavior. Let's talk about that just a little bit. Over 800 species are known to make sounds. I suspect that in the future we'll find that many more species, including those living in the great depths, make sound and use them for communication. The sounds are variable considerably. They're knocks, they're grunts. They're produced in a variety of different ways. As I'll show you in a minute and we'll play sounds for you, things like haddock, which are very well known, have a complex acoustic behavior that is involved with reproduction. And in fact, investigators have now learned that they can listen for the sounds of haddock and other species and find animals that are in breeding. Interestingly, many other species of fish do not make sounds. Something like the goldfish, while they hear very well, don't hear sounds, but we think and they don't make sounds to communicate, but we think what they're doing is getting information about their acoustic environment. The previous sound you heard was that produced by a haddock, and they produce sounds using the swim bladder with a series of drumming muscles that are on the walls of the swim bladder. This is typical of a lot of species. The exact pattern of these muscles are variable between species and even within the gatted fishes. In all cases, the muscles contract and produce the fundamental frequency of the sound, and the swim bladder serves as a way to amplify the sound, and that sound is detectable by other species, other fishes in the nearby area. This is an illustration showing you what some of the sounds look like from a haddock. This is what a male solitary display would look like, and you can see that there are a series of pulses that are produced by the contraction of the swim bladder muscles. This figure shows the spawning behavior of a haddock there with a male, the smaller animal, and the female, and they start out through a series of displays. At each point in the display, a different sound is produced. So it starts out at the bottom of the slide, goes to the top of the male and female together, and once they start spawning, the sound is stopped. So let's now switch from sound production and communication and talk something about fish hearing and how fish detect sound. But first, before going into some of the data that we've accumulated, I want to digress because the issue of what hearing is and how to measure it is something that's quite open to discussion at this point. Hearing is generally thought of as perception of sound by an animal and the ability to being able to behave or respond. In effect, it means that an animal has not only detected the sound, but it's been processed by the brain. Maybe various types of signal processing takes place, and the animal then responds. There have been two primary methods used to study fish hearing. The more classic method, first pioneered by Nobel Prize winner Carl von Frisch, was behavioral methodologies where fish are trained to respond to a sound when they detect it. The second measure, which is much more recently used, is based on what's known as ABR or AEP, which is a brainstem response that measures the output of either the ear or the brain during sound. The ABR or AEP method is recording at some point in the lower part of the auditory system either the ear, the eighth nerve, or the brainstem. It doesn't allow for processing of the signals by the brain or for the animal. The brain may actually extract signals in the presence of noise or determine things like sound source direction. Thus, ABR-AEP, while a very important tool, does not necessarily represent what the sounds the fish can hear, only what the ear can detect. As a consequence, what you can get is a sense of bandwidth, a sense of sensitivity, and it's a very useful technique when comparing between different animals that have been treated in one way or the other for things like hearing loss. But you cannot, from ABR or AEP, determine whether the fish can discriminate between sounds, whether it can detect signals in the presence of a noise or a master, whether it can determine sound source direction. In contrast, the behavioral approach to determining hearing is a mechanism whereby we actually ask the fish what it can hear. So it is how to use its whole brain to process the sound and then elicit a behavioral response. We've used a couple of different methodologies for training fish to do behavioral hearing studies. One of the early studies was shown in this figure, developed by Bill Tavalga and Jerry Wadinsky back in 1963, where they used a technique called a shuttle box, where the fish was put in a tank with a barrier. The sound was turned on. After a few seconds rather, a shock was given and the fish would swim across the barrier and learned that when they went across the barrier, the sound would go off. So after a while, the fish would avoid the shock by going across the barrier as soon as the sound was turned on. We can also use classical conditioning pioneered by Tony and by Dick Fay, where we measure changes in heart rate when the animal hears a sound. And we condition the fish to change its heart rate whenever it hears a sound to again indicate that it is detected. In each case, the animal is trained to respond. It only responds when it can detect the sound. If it does not respond, then we know the sound is not detected. We combine that with a psychophysical method, such as that shown in the lower part of the figure, called a staircase method that was pioneered by Tavalga and Wadinsky for fish hearing, where the sound is presented to the fish. It's lowered from after the animal's made a correct response until a point where it no longer shows a response. The sound was then raised until it detects the response, lowers and raises, and this threshold, which is defined as the detection 50% of the time, is in between the lowest sound level the fish can not detect, and the lowest one it can detect. And this provides a true behavioral measure of hearing. With this methodology, we can study masking, discrimination, and a whole range of other. A second digression is a very important issue that we've been raising for a number of years now, is where one does studies of fish hearing. The bulk of the studies, including many of the studies that I've done, have been done in laboratory tanks. The problem is there are significant issues as to the usefulness of those data because the sound field in tanks is extremely complex, generally much more complex than a fish would expect in the open water. It's very hard in tanks to measure particle motion. It's in all different directions. It's again unlike what happens in the wild, and so it's very hard to get good particle motion thresholds, which are really the things that we now know to be interested in because fishes detect particle motion and sometimes they detect pressure. We have developed in my lab, in Tony's lab, and a few other places, tanks that are useful that we can use to measure hearing, but these are often very big, very expensive, and very difficult to have built. There's also the issue with tank studies is that the differences in this data for the same fish species from different experimenters is very, very substantial. For example, Dick Fay first showed, and then he and Fritz Loddick showed it again, if you compare the data for goldfish from different laboratories determined behaviorally, their differences are rather remarkable. And it means that we really don't know exactly what the goldfish can hear in terms of thresholds particularly. And this is because the tank acoustics was so different from experiment to experiment. The conclusion you have to come to is the most effective way to get hearing data is in field studies, as something like shown in this figure that from work Tony and Hawkins did with colleagues in Loch Torridon in Scotland, where the animal is in a deep water lake in Loch. The animal is in a cage. Speakers are placed at different distances some quite substantial from the animal, and fish hearing can be measured in an acoustic environment that is very pristine, where there are no reflections, and where we can actually calibrate the sound field. So now let's talk about what fishes can hear. This figure shows several different points of considerable interest. First of all, the x-axis is frequency, the y-axis is the lowest sound level detectable, and it represents behavioral data from a variety of different species. The significant thing is to show the tremendous variability in sensitivity, that is the lower number that the fish can detect, and bandwidth. You can see there are some fish like the perch that don't hear very well. Animals like the grunt and damselfish and squirrelfish hear a bit better, and goldfish and soldierfish hear quite well, up to 3,000 to 4,000 hertz. The significant things here are that, first of all, the European perch only detects particle motion. We suspect that the grunt and the damselfish and the squirrelfish detect particle motion and perhaps pressure, whereas the goldfish and soldierfish both have unique specializations in the auditory system that enhance their ability to detect sound pressure and broaden the bandwidth and increase sensitivity. What's also interesting is that the squirrelfish and soldierfish shown here, both animals studied by my former student Cheryl Coombs when she was my graduate student. Cheryl is now retired, so you can determine how long ago that might have been. Those species are sympatric species. They live very close to each other, sometimes in the same caves. They're closely related, and yet if you look at the ear and the connections to the swim bladder, they're very different in the two species. Both actually make similar sounds. So the hearing in fishes is very, very. The best studies looking at sound pressure and particle motion were work done by Tony Hawkins and his colleagues, and this slide shows some of those data, work done in the field. In the case of the dab and the salmon, both species that were here only to low frequencies, they're only particle motion detecting fish. The cod is an animal that detects both sound pressure and particle motion, and the herring is a species that's adapted to hearing higher frequencies by having specialized structures in the ear that enhance hearing. Interestingly, some species related to herring in the same group, the members of the group Alocinae, including American cod, can hear actually out to 100 kilohertz or more to detect dolphin sounds and avoid being eaten. But these have high degrees of specialization found in few other species. So now we know the fishes hear, there's variability, and the question comes up is, how do fishes detect sound? Well, this is an image that was drawn by Carl von Frisch back in the 30s. What he shows here is the head of a minnow, a European minnow, with the side of the head taken away. You can see the brain, and you can see in yellow the ear of the species, and you'll see that the ear is in the brain cavity. I will tell you there's no external ear in fishes. There's no middle ear. They're not needed. And some fishes have ancillary structures I'll talk about later that help them detect sound pressure, including this species. But this is a typical location of a fish ear. This is the drawing done by Tony Hawkins from work done by Toradol, showing the ear of Atlantic cod. And you can see the brain, you can see the ear. The ear in fishes have 3 semicircular canals like we do. There are 3 otolith organs, the saccule, the utricle, and the lagina. And the ear is typical of most species of fish. The otolith organs, which are involved in hearing, each have a dense calcareous otolith, which you can see in the picture, and a sensory epithelium, they're next to one another. And as I'll show you in a minute, the hearing is related to the relative motion of the very dense otolith and the epithelium. Show you another picture. Here is an otolith lying near the sensory epithelium. You see the sensory hair cells and the 8th cranial nerve that projects to the brain. Each of the sensory hair cells has on its apical surface a ciliary bundle, and these ciliary bundles can be bent, and I'll show you that in a second. So this shows you the ear of a typical fish. The lower image shows you the whole sensory epithelium. It's shaped something like a sperm with the anterior end to the right. If you magnify that, you see the ciliary bundles on the sensory hair cells. These project up into the lumen, and the image on the right shows you a cross-section that demonstrate this is a typical vertebrate hair cell. It's not very different than the sensory hair cells with which you're listening to this talk. This image shows you the otolith on the left and the sensory epithelium on the right. And the point we want to make is that they are connected by what's called an otolithic membrane. It's a gelatinous mass that we don't really know the structure of because it's very hard to work with, but it seems to connect the otolith to the epithelium. So when the relative motion takes place between them, the otolith cannot go too far. Let's talk a bit about how fish hear. The fish that I said are detected as a particle in motion. As the sound comes along, the fish's body moves in phase with the water because it's the same density as water. The otolith, being much denser, lags behind the motion, and because they're connected by that otolith membrane I showed you a moment ago, the cilia are bent. This causes release of neurotransmitter, and the animal detects the sound. As I'll show you in a minute, some species can also detect sound pressure by having the presence of an air bubble, which is in most cases a swim bladder, which re-radiates particle motion and stimulates the sensory epithelium. So there are several different pathways of sound to the ear that take advantage of pressure and particle motion. As shown in figure A, there are many fishes, including elasmobranchs, that have no air bubble, and those only detect particle motion in what we call the direct method of stimulation. The stimulation is that I showed you in the last slide, with the sound particle motion rather, stimulates the fish's body relative to the otolith, and you get stimulation of the ear. In some cases, the fishes have a swim bladder at some distance from the ear. These swim bladders detects pressure, re-radiates energy, but since there's a substantial distance between the swim bladder and the inner ear, the particle motion that's re-radiated attenuates quickly and is not detectable by the ear. However, in some species like the cod and the soldier fish I showed you a little while ago, the swim bladder has anterior projections and it ends close to the ear. When it re-radiates sound, the particle motion is substantial enough that it too can stimulate the ear. As a result, these fishes, the ones that have a swim bladder near the ear, or actually may have a bubble of air in the ear itself, these animals detect sound pressure as well as particle motion. To show you what this means for a fish, look at this slide. It shows you frequency on the x-axis and the displacement amplitude that would stimulate the ear on the y-axis. Focusing on the green line, if a fish just has a particle motion detector, the signal level decreases at higher frequencies. So it starts out at low frequencies, the displacement may stimulate the ear a lot, and as it goes to higher frequencies, detection gets poorer. However, with the presence of swim bladder, there's this increase in sound pressure, which increases particle motion, which effectively means that the bandwidth of particle motion getting to the ear is much wider rather. This increases the bandwidth and the sensitivity of the fish over a much wider range of frequencies. So the swim bladder actually allows the fish to detect particle motion at much higher frequencies. One of the most striking features about the auditory system of fishes is the striking interspecific variability. And consider that there are 33,000 or more bony fishes in the world. The variability in ear structure, particularly as related to the overall ear shape and the shape of the otolith organs, is rather dramatic. In fact, it's far greater variability than in any other vertebrate group. These differences include ear shape, configuration and shape of the otoliths, the shape and configuration of the sensory epithelium, the lengths of the cilia on the hair cells, and many other features of the ear. So this shows you a number of actually related species that work by Xiaohong Deng, showing you the tremendous variability in the shape of the ear and the otoliths. This figure shows you the variability in the otoliths themselves from different species. What you can see is gigantic differences. We have very little understanding of what the shape differences mean. Fisheries biologists use otoliths to age fish. They put down rings every year, but why the shapes are there is something that we still ponder, although I think the general consensus is that it's involved in various aspects. So there are two fundamental questions about this variation. One is, what does the variation mean? Do the differences reflect different methods of extracting information from the sound, that the fishes use different aspects of the sound for hearing and pay attention to different things? Or alternatively, are the different adaptations just different ways that have evolved the fishes in different groups to extract the same kind of information? At this time, we have no answer to these questions, but this becomes one of the most fascinating and important questions in fish bioacoustic studies today. So in summary, what do we know about fish hearing? They detect particle motion, some detect pressure. Their ears are actually very similar to ours, and hearing evolved in fishes. The ear evolved in fishes. The sensory hair cells are very similar to those in humans. And like other vertebrates, particularly mammals, fishes detect a wide range of frequencies. They can discriminate between frequencies almost as well as we can. They can discriminate between intensities almost as well as we can. They can detect signals in the presence of other sounds, or other sounds are called maskers, and they can determine the direction of sound sources. These are all characteristics in the auditory system that allows the animal to use the acoustic scene to understand its environment. But there are many open questions. The mechanics of the ear are things that fascinate many of us, and it's a very difficult problem to get at. Sound source localization, which the first studies were done by von Frisch and Dycroft in the 30s, is still an enigma for us. We still don't understand the mechanisms of discrimination and the comparative issues. The variation between different species is a fascinating project that really is open to study. So as I've indicated already, Tony and I have been working on fish hearing for many decades, but over the last 15 or 20 years, we both got very involved and interested in the effect of anthropogenic sound on fishes. So for the remainder of the talk, we'll consider that as an issue. This is an image that was painted by the very great maritime artist Turner, and it represents a massive change in ocean acoustics. That is, up till the early 1800s, the only sounds in the ocean were those naturally produced by fishes, by marine mammals, by geologic events. Starting in the early 1800s, with the advent of steam transportation, the amount of noise produced in the oceans and bodies of water by human activity increased dramatically. And this picture shows one of the first steam engines towing one of the last sailing vessels. But this increase in sound in the ocean environment and the lakes and rivers has had potentially have a dramatic effect on marine animals. Why is anthropogenic sound of such concern? Well, basically, we've already demonstrated that sound is very important to aquatic life. Anything that impairs the detection of sounds can affect the fitness and survival of aquatic life, and thus we have this growing concern about the potential effects. Now, the initial studies on the effect of sound were started with marine mammals, and this continues, of course, and it's a very important issue to consider. But more recently, the concern has spread to fishes and invertebrates. Clearly, they are major parts of the human and even marine mammal food chain, and anything that impacts the fishing capabilities or the efficient survival of fishes can have a dramatic impact on humans. There's also concern about marine reptiles and birds. I think there's a talk at this meeting about the potential effect on birds. There's considerable concern about the effect on turtles. This figure gives some sense of the types of potential effects and where they're located, and this all depends on the received level of the sound and the nature of each individual sound. Very close to a very loud sound source, fishes may be killed. This doesn't happen very often, and there have been very few experiments to confirm this. A little bit up to greater distances, there may be physiological effects that I'll talk about later on. There may be temporary loss of hearing. Up to greater distance, there may be masking of sounds, so fishes don't detect sound as well as they could. And even up to greater distances, there may be various behavioral effects. So the potential effects can be, as I said, include masking. They can mask communication sounds or soundscape. They may cause hearing loss that's temporary. There's no evidence of permanent hearing loss in fishes. Fishes may be driven from feeding sites or spawning sites temporarily or permanently. They may affect, sounds may affect fish migration routes. They may damage body tissues with the sounds are very intense. Very intense sound may kill fish. In all cases, may affect fitness of individuals and most importantly of populations. So there are many sources of anthropogenic sounds that could affect fish. This figure just gives some sense of them. There's pile driving, wind farms, and so forth. And I'll show you more of this in just a few seconds. This figure demonstrates sounds from shipping and boats. These have become much greater over the past decades, and there's no reason to think this won't continue increasing. Bodies of water like the Chesapeake Bay, which is near where I'm located, rivers where there's lots of shipping and the shipping is increasing, we can imagine that the background noise is growing tremendously, also in shipping lanes, and this may be driving fishes from spawning grounds, or it may prevent hearing of fish calls, and it's a constant sound. Pile driving sounds have become greater, as I'll show you in a few minutes, mostly from things like marine construction, particularly of wind farms and harbors. Pile driving not only produces sound in the water, but it also produces sound in the substrate, which may get out of the water or affect animals living in the substrate. These sounds are extremely loud, or can be depending on the pile being driven. They are probably as loud or as those produced by the next thing I'll talk about, which is seismic air guns, and the sounds are impulsive. They bang, 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 and they can disrupt behavior and do masking. But what's interesting about them is they don't necessarily be continuous. They may happen for a couple of days or weeks, and then they'll pile driving sounds, as I said, are impulsive, and they can be extremely loud. These sounds can be produced particularly in construction of offshore wind farms, and this is becoming a big issue in many parts of the world. There are two problems with wind farms. One is construction noise, which is produced by pile driving, and the other, which we know much less about, is the operation of the wind farms, which continues to generate substrate vibrations and low levels of sound in the water. And there is concern that this may affect fishes and other animals. Interestingly enough, in many places where wind farms have been constructed, there's a massive attraction of fish that seem to like to use the wind farms and their structures as places to live. So whether the sounds are bothering them or not, it still seems to attract lots of fishes to their locales. Seismic air guns are again a very large issue. And they are impulse sounds. I'm sorry, the sound didn't work. They are used for oil and gas exploration. The sounds are very loud. They produce not only sound in the water, but they intentionally produce sound in the substrate, and that's necessary for the exploration for oil and gas. These sounds go on for hours and days in the same area, potentially disrupting the behavior of the animals nearby. The next figure shows an actual experiment that Tony Hawkins was involved in, where they explored the pelagic group of fish, whiting, which are related to the cod. They live in mid-water, and they were observed during the experiment where a boat was producing sounds from a seismic air gun over a loudspeaker. What you can see is a school of whiting with the darks on the left. They were at a certain depth, and as soon as the air gun went down, the fishes went considerably deeper and stayed there until after this air gun stopped going. So they were clearly disturbed by the sound of the air gun. Work in Norway over the last 20 years or so has shown that if you measure fish catch of a number of different species, as soon as the seismic study starts, the fishes leave the area or go to great depth, only to return when the sound has gone off and been off for several days. So while we have a good sense that anthropogenic sounds can have an impact on fishes, there are still many questions about all kinds of aspects of this. The trouble is there are very few data on the effects of sound on fishes, and there are a number of reasons for this. First of all, there's been very poor funding for work on this area. Most of the money on research on anthropogenic sounds has gone to studies of marine mammals. Much of the work has been done in the laboratory by various investigators. The trouble is, as Tony and I have shown in a number of cases now, data from the laboratory study on the effect of sound on fishes cannot easily be transferred to the field where the animals live because of the strangeness of the acoustic environment of a fish tank and the fact that the sound fields are not typical of animals in the wild, plus the fact that animals in cages or tanks tend to behave differently than they do in the wild. Extrapolating from sounds in the lab to the field is very difficult and highly problematic. The other part of it, though, is that it's very hard to do field work to observe potential effects. So while the data from labs are not applicable to the field necessarily, doing work in the field is very hard to do. And adding to that is just calibration of the sound fields in the field is also difficult, and determining the actual stimulus that affects fishes is difficult. So there are many issues that make doing studies on the effect of sound on fishes very hard. So what I want to do now is briefly talk about two projects, one that I conducted in my laboratory over several years, working with my postdocs, Michelle Halverson and Brandon Casper, and then we'll talk briefly about a study that Tony has done recently that has been very revealing, and a field study. My lab has been investigating the effect of pile driving on fishes, and we realized quickly that we could not do work in the field near a pile driver. They're dangerous, and you can't control a pile driving operation that costs tens of thousands of dollars for each pile being driven. So what we did was invented a way to bring a pile driving sound into the laboratory by developing a special device we called a hissy and in there we could produce sounds that were very similar to those produced during pile driving at the same levels, and then we could explore what a potential effect these sounds could have on physiology and physical nature of the fishes. We were able to do a variety of different experiments to ask these questions. So our general approach was to place fish in the structure we call the hissy fit. It's a tank that has two very substantial speakers on either end. You see the open hissy fit here. We've closed after the fish were placed in there. The tank was closed and the sounds were generated. To give you a sense of how loud these sounds were, the hissy fit device was on the third floor of a steel building. And when we first did it, we didn't have enough vibration isolation. The whole building shook every time we played the sound. We quickly figured out a way to isolate it from the building. Otherwise, we would have been kicked out. We've exposed fishes to sounds with varying numbers of strikes and sound levels. After the fish were exposed, we removed them. We examined the fish for barotrauma or injuries, and we used necropsy procedures, and then we held some fish for post-exposure. Just as a quick digression on sound, what we were concerned with was not the things that people normally are concerned with sound, that's peak sound level or RMS sound level, but we're concerned with sound exposure level, or the total energy within each strike. So we dealt with what's called the sound exposure level, or SEL, for either a single strike, which is SEL-SS, or we accumulated the sounds, which is the most important issue. How much sound is a fish exposed to over 5, 10, 100 different impulses from a pile driving apparatus, which we call the cumulative SEL. So as I said, we exposed fish to either 960 or 1920 strikes. We varied the sound exposure levels, and we did studies in a variety of different species to look at the effect on body tissues and see how that changed with different species. Just to show you what the injuries look like, we had mild injuries, things like damage to the eye, a little bleeding in the eye or the fin. We had moderate injuries internally, which included hemorrhaging of the liver or bruising of the swim bladder. And then we had what we call mortal injuries, which were internal hemorrhaging, damage to the swim bladder, damage to the kidney. Interestingly enough, the major damage we saw only came from the very loudest sounds and only occurred when the structures were near the swim bladder. You'll give you a sense of the kind of data we got. This shows you a graph of the number of injuries at different sound exposure levels. On the left, you find animals that were exposed to the lowest levels. They may have had just 1 or 2 injuries. As the sound exposure level increased to the right, what we saw were many more types of injuries and many more of them in every individual fish. So what we were able to do is get some kind of graph showing that as you increase the sound exposure level to which fish encountered, the number of injuries occurred. Now, keep in mind, these were very loud sounds, extremely loud, and equivalent to the fish being very close to the sound. So lived the sounds. So what we found from our studies was that the onset of physiological effects in fishes, that is some kind of damage that may have been significant, occurred with a cumulative sound exposure level of 207 to 210 dB. It varied by species. Mortality does not occur until the sound exposure level is substantially higher at 215 dB cumulative sound exposure level. We found the effects are generally the same between species with very different morphologies, but there were distinct species differences. There was no effect, interestingly, in fishes that do not have a swim bladder, supporting the hypothesis that the swim bladder motion during very intense impulsive sounds is what causes the damage. We did recovery studies and showed that the non-mortal injuries recovered in less than 10 days, but with a caveat on this is these were animals kept in the lab, they were fed, there were no predation. Animals in the wild with the same damage very likely would have been preyed upon and eaten and probably could not have fed. We had no injuries post-exposure, and as anyone would think about, would ask what happens to the ear. It turns out ear damage only took place at levels well above those that started to show other injuries. And our thinking is the ear in most species is not near the swim bladder, so damage does not occur. So a second study is done by Tony Hawkins with colleagues in a loch in Scotland, in Ireland, and what he did was to put down a sound source and use an echo sounder to watch the behavior of fishes during stimulation from this sound source that was playing back pile driving sounds. It's a great study because it was done in the field where many aspects of the signal were controlled and the recordings were controlled. And he looked at the effect of sprat on sprat and mackerel, and what he found in essence is that both species during playback would change their behavior, particularly from. In this experiment, Tony used a variety of different sound levels, and the sounds were transmitted at different levels. They were able to determine which sound levels caused at least half the population of groups of fish to respond. They found the sprats were sensitive to sound pressure, and more than 50% of the sprats responded to sound pressure levels of 163 decibels. In the case of the mackerel, which don't have swim bladders and are only sensitive to particle motion, about 50% of their schools respond to a particle velocity level of minus 80 dB. And this occurred when the sound pressure was at 163 dB. These sound levels often occur at distances of several hundred meters from the actual pile driver, so pile driving can potentially affect fish over a wide area. And these are the first 2 species that have actually been examined in the field in response to equivalent to pile driving sounds. Interestingly enough, in the same study, Tony observed zooplankton, which is the food of the 2 species, and he found they respond to the playbacks of the pile driving sounds by moving deeper in the water. The zooplankton consisted mostly of copepods and crab larvae and other species eaten by the pelagic fishes, including sprats and mackerel. And so what happens is not only we are potentially affecting the fishes, but also the food that they eat. As I indicated earlier, we know very little about the effect of anthropogenic sound, and this just gives you a list of some of the major research questions that need to be explored in the future, including effect on different species, studies on sensitivity of fish to different sounds. They need to focus on particle motion, affected different types of anthropogenic sounds, the effect of different types of sources, behavioral response to sound in wild animals in the field, and things like the contributions of substrate vibration to anthropogenic effects. All of this work is involved with the issue of regulatory issues and the setting criteria for use of anthropogenic sounds in the presence of fishes. The question really comes down to being, how do we set criteria that regulators can use that allows studies and work using anthropogenic sound without harming the animals and without affecting the populations? It's critical, in our view, that any criteria must include behavioral and physiological effects, and they must take into consideration variables as things like intensity of signal, whether the sounds are continuous or impulsive, signal duration, and a variety of other issues. In other words, there's no one criteria for anything that can be used for all animals. The problem is the current criteria in the world, there are very few of them, are very much one criteria fits all. The best known criteria were developed by US agencies in 2008. They were not based on the best available sciences, nor have they been updated. However, in 2014, an international working group, of which Tony and I were very much involved, developed interim criteria that are now being used, at least informally, in many parts of the world. And unlike the previous criteria developed in 2008 in the US, this takes into consideration different species groups, different sound sources, and different types of effects. And the work is based on the best available science at the time we did the work. Tony and I, along with Michelle Halverson, recently updated the literature and came to the conclusion that the criteria and guidelines we set in 2014 are still very relevant today. This just shows you what the criteria look like. As I say, it was developed for fishes. We also did work with turtles. They are still only in terms of sound pressure and not particle motion. We also need criteria for invertebrates, and ultimately we will need data and criteria for substrate vibration and its effect on animals. This just shows you what the criteria look like. I'm not going to go through this, but each of the criteria we set for different sources were based not only on the source type, but also on the different types of impairment, which is across the top, and the different animal groups down the bottom. And we set, based on the best science that were available at that time, we set criteria that were considered to be interim, but at the same time, you'll see that many boxes are not filled in because there were not the data to provide the information that we need. Finally, let me just end by saying that in conclusion, what we've shown you is fishes have the same basic hearing mechanisms and capabilities of other vertebrates, including mammals. Anthropogenic sounds is a very large issue around the world. Far more research is needed in order to develop the best ways to protect fishes from anthropogenic sound. But it's very important to note, not all anthropogenic sound is bad. It's not good either, but it's not bad. Keep in mind that humans and other animals function in a world with all kinds of sound around us, both natural sound and man-made sound. In general, most of these anthropogenic sounds around us don't bother us. Highways, roadways in front of us, things of this sort, we adapt to these things, and it's very likely that fishes are not affected by many of the sounds. They ignore the sounds or habituate to them. However, we need to understand this issue better, and much more research is needed. So this just gives you a number of our recent relevant papers that Tony and I worked on. You're welcome to email us if you want copies of these or the other papers we've done. And finally, permit me to mention that in 2022, we are going to have the 5th Effective Noise on Aquatic Life meeting in Berlin, Germany. These meetings, which have taken place every 3 years, have become the place to go for meeting with regulators, scientists, industry, all interested in the effect of sound on aquatic life. And we'd be delighted if you would consider joining us. Thank you very much for your attention, and we appreciate being able to be here today.
Okay, so I'll kick off then. So I just want to say a massive thank you to you and to Tony for putting the presentation together. In my run-in to introducing your plenary earlier today, I did say how great it was that running this conference, you get to email people and people who are absolutely amazing and so proficient in their professions, and then, you know, have their enthusiastic response. So I'm very, very happy that you agreed to put the plenary together. And it was really a very necessary plenary, I think. I know that we'll be referring back to that. I'm actually going to talk to you about whether we can make it more available as well, perhaps on a YouTube platform or something, because the background information that you guys provided, I think, is relevant to so many people working here. And particularly, just to say, you know, in my own sort of conversations with policymakers here, we often get questions about, okay, well, what's happening with noise? How are the fish affected, particularly with seismic, and also construction like pile driving and things like that. So the aspects that you covered so eloquently, I think, are going to be very, very useful for those people who are making decisions about the effects of noise.
Well, that was one of our goals. That's been, you know, part of the goal of things that Tony and I have been doing for the last couple of years. Is you can talk to scientists a lot, but they're not the ones making the rules and setting the regulations, and we need to be contributing.
No, it was brilliantly done. So thank you so much.
Thank you. That's very kind of you to say that.
Well, no, I was I was absolutely thrilled.
Well, no, I appreciate it. It's nice to hear.
Yeah, no, it was, and there is so much sort of mystery about it. And I mean, we've got obviously quite different species here as well, and so obviously different species all have different hearing thresholds and perhaps different, you know, behavioural responses as well. So I would love to see, and I know many people in the African Bioacoustics community would love to see further research being done on this. I was going to say school of research, which I'm coming out with all the fish. All the fish puns today, but I think having that background information then also helps to inspire people to research more into that. We are having a question, I think. Is that right? Yeah. Okie dokes. Okay, so we've got a few extra questions coming in right now. I'm going to push ahead without Arthur, which is sad, but without Tony. But in the interest of time, I guess that's what we'll do.
Let's go ahead.
Okie doke. So the first question that we have through, the way that the questions work is that people pose them on the online platform, and then they get voted. So it's a little bit interactive.
They get what?
They get voted.
Okay.
Right. So actually, I should say to everybody listening in right now that there's still time to post questions. We've got questions up until 6 o'clock. And you can still vote for your top questions as well. But as soon as I read the question, it's answered, then those gets marked as answered. So that's how it runs. Okay, so the top question we have for you here is, have you observed a trend in hearing abilities depending on the fish habitat? So, for example, benthic versus pelagic depth in the water column.
A super question. What's the name? I think it was Hans Slavikorn and I wrote a paper about that a number of years ago, or somebody else. The answer is no. And part of the problem is that there are so many species of fish, and we have good hearing data on so few, that making any generalizations like that would be virtually impossible at this stage. Again, consider that 33,000 species or more. We have good data on 3 or 4, and I say good data. We have sound particle motion data, we have sound pressure data, and extrapolation is impossible at this stage of the game. And my guess, is strictly a guess, is the variability is related to other factors. The species I think of is squirrel fishes, holocentrids, where you have 2 species that live together. They look alike, but their hearing capability and their sounds are similar, but their hearing capabilities are totally different.
Yeah.
And they're both living on a reef. So, you know, at this stage of the game, generalizations are probably impossible. The only thing I can say, and this is very much a guess, is that deep sea fishes, many of the ones we've looked at, and this is mostly by a former student of mine, Xiaohang Deng, when she did really phenomenal microscopy, they seem to have a lot of species with specializations for hearing. Mm-hmm. And they may hear, but again, even within taxonomic families there that live near each other, there's tremendous variability in the ear. So I don't have an answer. I don't know. It's great, but you know, it's worth at least 3 or 4 doctoral dissertations.
Yes, exactly. And I mean, actually, it sort of relates to the next question, which is, you mentioned the difference in the otolith form. So
Right.
the different otolith shape. And so, yeah, I mean, this was my question actually was, do taxonomically similar species have similar shapes? Or does it more depend on the environment they operate in? So similar to the acoustic adaptation hypothesis. And so then do you get convergence in otolith shape between species which have similar ecology, but perhaps are not taxonomically related? But I think that also feeds into the question that we've just asked.
Yeah, that's a good question. I would guess the person to ask would be someone who looks at otoliths a lot more than I do and correlates with ecology. There's groups in all over the world because, as you know, otoliths are used by fisheries biologists for aging and things of this sort. I've seen, going back to some work that my student Xiao Hong did on melampids, within one group, the otoliths totally varies from pretty mundane to things that we've never seen before. One species actually has, and we use the word very casually, a spear sticking out of its end, doing something we don't know about, but it's as long as the whole otolith. So, and that's within one group. So, and they're all a deep sea fish. So the ecology is probably fairly similar. So again, I don't think there's a correlation at this stage again one can make. I think the diversity of fishes is so great that any correlation at this stage of the game would have to be very, very tenuous. But again, it's a great question that we need for which we need a lot more data to try to answer.
Yeah, I mean that's it. You've got so many different species and different habitats. Right. You have to know the habitat that they're in and their ecological role before you can even get to that question. And then you need to feel out, well, is that actually functioning in the different hearing?
That's it. We just don't know at this stage of the game how the ear works in fishes. We have a general sense, and we talked about that. But why do you have this variability in form? And that is really, from my perspective, the most interesting question to ask. Yeah. But doing the experiments are very, very, very difficult. And modeling is very difficult at this stage. So it's just, there are more questions than answers. Far more, which is good science, of course.
Yeah. There's a lot of work to be done.
Yep.
Okay, well, thank you for that answer. I'm going to move on to Guy, who asks, nice talk. Congratulations. I have a question. Do you think there could be potentially another source for this for sound mechanic wave reception instead of the ear? So in other words, could catfishes and other, he's put in a difficult word there. I can't speak, it's very late in the day.
Siluriforms?
Thank you.
What was that?
Siluriforms?
Oh, those are catfish. Okay, it's siluriforms. It's an order of catfish.
My being able to read is pretty poor right now. So could catfish perceive sounds by the mustache, via the trigeminal nerve, or for most fishes?
Trigeminal nerve.
Sorry, my pronunciation.
That's okay. Obviously, you're not a fish biologist, so that's understandable. Well, first is asking a couple of questions there that may not even realize they're asking. What is hearing is the first question. And they may be detecting water motion with the barbels, but sound itself is sort of defined as something that's detected by an ear. And so the answer would be no, but it doesn't mean that they aren't detecting vibrations and stuff that contact the barbels. There was a study years and years ago by someone in Denmark, whose name I cannot think of at the moment, and he found that codfish could detect sounds up to about 40,000 hertz. And after a while, what he realized is that it wasn't the ear that was detecting it, but the animal was detecting vibration by pain touch receptors, we think. And so it was detecting, but it couldn't do anything with the sounds. It was just, it was there, and it had to be extremely intense, much more intense than would normally happen. And so it was using a different sense, but it's like, years ago, there was a gentleman named George von Beckersey, and you probably never heard of him, but he won the Nobel Prize in 1960 for studies of hearing. And one of the things he did was to try to understand how the cochlea works in the inner ear. And what he did was develop a methodology where he had a long piece of plastic that was shaped like the cochlea. I don't have to go into details. And he put it along his arm. And if you put your arm on there and he changed frequencies, you'd feel it along your arm. So essentially you could hear the mechanism of the ear on your arm, but it was it was all touch receptors. It wasn't hearing, it was touch, but it could mimic the mechanics of the ear. So what I'm saying is that, and if people don't know Beckersey's work, it's phenomenal. It's clear why he won the Nobel Prize. He was also a nice man. But what was that?
Which is very important. We were talking
yes,
about
he was our neighbor when we lived in Hawaii. But the point is, getting back to the catfish, it's possible that it's sensing things, but it's not hearing, it's touch or something else.
Yeah. You know we're all going to go home tonight and put like things on our arms and try this out, right?
Well, think about this. If you're sitting around and a bug walks along your arm, you're feeling it with touch receptors or the hairs on your arm. That's the same thing that Beckersey was doing. But that's not hearing. That's touch reception.
Sure. Amazing. No, that's cool. And he got the Nobel Prize for that work. I mean,
he got Nobel. Well, what he did, he figured out how basic mechanisms of hearing.
Yeah.
So,
brilliant.
Great man, great intellect.
All right, we're all going to be also Googling more and more now as well. We should have probably done it before we started.
I make all my graduate students learn who he was and understand what his work was.
Sure. Okay, I'm gonna move on to the next question. This is really fascinating. And the question is, what are the telltale signs of acoustic impact causing injury in fish? So can non-specialists pick this up? And this has relevance, I think, in terms of, people. I had it actually yesterday. Somebody sent me photographs of a whole heap of dead fish in a river where we live, and basically said, what caused this? And these are the sorts of questions that people want to know the answers to.
Yeah, that's an interesting question. I guess we see that a lot. I was involved in a project on the Hudson River in New York City with building a very large bridge. And the concern there was the sounds from pile driving affecting sturgeon, which is an endangered species on the US East Coast. And the problem is you get fish coming up dead, but it was very hard to determine if the death was related to the pile driving or to something else, because they look alike. It's a burst swim bladder maybe due to something, who knows what it's due to. And so unless you can correlate the damage to an incident where there's been intense sound, it's very hard to discriminate. Also, a lot of times you get dead fish and they just died and the tissue starts to degenerate. It's very hard to tell. So there's no simple way, and there's so many other factors could be in play that I don't, it's the same thing as, the issues with sound harming whales or squid. Unless you can really correlate the effect with a sound source, how do you know it wasn't something else? It's very hard to say, because a lot of the effects you see might be due to other things or just normal mortality.
Well, that's it. I mean, we get asked this question a lot. We do a fair amount of necropsy work here. And I always feel like, if we say, well, we're not sure, it could be cumulative, it might be this, it might be that. But I also know that, I don't have a detailed veterinary background or, the skill set necessarily to be able to rule out one aspect from another discreetly. And so it's interesting to know, what you're saying about the different possible causes, for example, of bursting a swim bladder or something like that. I mean, I wouldn't even know what a burst swim bladder would look like. So yeah, I wouldn't even know where to start really. But I mean, I guess that you showed up pictures of sort of bleeding around the eye and things like that.
Yeah, that's right.
It could also be perhaps toxins or something that would cause.
But also, you know, it could be the fish went up to the surface really fast.
Yeah.
From a depth, or it could be or is hacked by another fish. It's just too many variables, especially external things. So we're doing some work now where, you know, we're seeing external damage to fish after exposure to an explosion. But what we're also learning is that a lot of the same things are in the controls, and it's just the normal pattern for the fish. So it's really hard to extrapolate unless you can, you know, there's a one-to-one relationship between the presence of the damage and a source that might have affected it.
Sure. And this is a related question, but it wasn't one that came in, but it's just sort of coming to me now. I mean, in terms of doing this sort of work, ethically, you know, how does that work in practice? Because if you're doing sort of playbacks of sound to fish, you know, and that has an extreme response or extreme outcome, what's the process there? I mean, how obviously, you know, there's a level where there's a balance between justifiable work, but also that's quite a difficult line to tread. And what's your thoughts on that?
No, it is, and you know, everything we've ever done, and I trust that just like you, any animal work has to be approved by an animal research board. In our case, what's called an IACUC, an Institutional Animal Care and Use Committee, and they're very rigorous. And what you can justify because these data are needed. At the same time, you have to work very hard to make sure you use minimal number of animals and you subject them to the least trauma you can. You can't do things like anesthetize them in advance because that affects the physiology. But you know, as you say, it's a trade-off. But we try very hard to make sure that we are exceeding the rules in protecting the animals and using minimal number of animals. But at the same time, at least in the States, the expectation as part of approval is you have to have enough animals to be able to statistically demonstrate that the results are valid. They don't want you just doing one animal and you're wasting an animal for all practical purposes then. So you have to develop a statistical protocol. And in our case, we always work with some very, very sophisticated statisticians to design the experiments to make sure that we're using the right number of animals, but not too many.
Okay, yeah, that's very interesting. Okay, I'm gonna move on and say, okay, we've got one question. So a couple of people sort of touched on this, and I'd be really interested to know your thoughts on this point. So, and this question is from Suan, but yeah, there was a related point I think in the chat, so I think it covers it here. And the question is, we know from studies in humans that they often, even when they don't feel that they've been impacted negatively by noise pollution, so that people live in airports so they don't notice the noise anymore, but nevertheless, they show some sort of physiological stress response, such as elevated heart rate, cortisol levels during noisy events. And so might fish and have similar sort of physiological effects, even if they look behaviorally normal?
Absolutely. We like to argue that whatever happens to you and me in terms of stress response from noise happens in all animals. So I would expect that a fish in an environment that gets really noisy because they've increased shipping very substantially may be stressed, and the stress will be the same things as we'd see, changes in hormone levels, changes in all kinds of things, sleep deprivation if they sleep. And we're arguing that the model system can be humans because the same things, we think the same things. There are not much data for fish to show this. But wherever there's been experiments to test it, it's clear that this can happen. So we need more data, but it's still totally be expected.
Yeah. And I guess that passive acoustics might be another way to look into that. So looking at call rates or something like that actually sort of might come full circle in terms of looking at their calling behavior relative to.
Well, it depends on if that's you do see change in calling rates with sound exposure, with the presence of other sounds. A friend of mine, Joe Liskowitz in North Carolina, has looked at call rates in some fishes that when dolphins are nearby, echolocating or looking for making sounds, and they change the call rate. So yeah, but call rates may not be indicative of a stress response, which could have long-term effects on health and well-being. That is, if you look at people even that are exposed to long-term sounds, even though this person has, you're not aware of it, it may still be effects on the endocrine system, on reproduction, on sleep deprivation, which have long-term health and well-being effects.
Yeah. How do people look into things like heart rate in a fish? I mean, how would you even begin to do that?
You measure it. Do an EKG. Why not?
They're in the water.
I suspect there are for a large fish. You could do it for a whale, can't you?
Oh,
a little bit more challenging.
Yeah, it's challenging, but at the same time, if you had the right size receiver or transmitter, I don't know anyone who's done it, but my guess is a colleague of mine, Barbara Block in Stanford. If it's been done, she's figured it out. They took long-term recording on sharks and tunas and things of this sort.
Yeah.
And yeah. I assume it's doable because putting an electrode in the recording of heart rate, that's easy, and we do that all the time. What we're doing is we're using heart rate conditioning to measure hearing, and you look at change of the heart rate when the animal hears a sound. You train it to change its heart rate, but that's doable.
Cool.
I think I wanted to ask, very random. Do you think? So we have someone called Sound on the participant list, and I'm not sure if that's Tony. I don't suppose if you know what his Zoom profile name is.
I haven't the slightest idea.
You could take the risk and let Sound into the chat because he's put his hand up, or her hand up.
Go on, let him in.
Yeah,
or her.
Her in. Sound?
Let's see.
Let's see. The risk, I know. I'm asking you to unmute.
No.
Hello?
Hello, can you hear me?
That's Tony.
Tony, hi.
Yes, it's certainly Tony, but I can't understand. I can see all of you. I'm not sure why you can't see me or hear me.
We can hear you, Tony.
We can hear you now.
Okay, you can hear me, but you can't see me.
We're bringing you in so you can put your camera on now. To be able to put it on. Sorry about that.
Oh, it says sound in the middle of the screen now.
That's what your name was, so we couldn't find you.
That's my name, I guess. So, but I still can't see. The host has asked me to start my video. So I'll start my video.
There we go.
Yes.
There you are.
You can see me now.
Wonderful. That's so cool.
Yeah.
Right, I think Art's given you very good answers to all your questions so far.
I was gonna ask you if I was okay with you, Tony.
Yeah. Yeah.
Well, I'm glad
Well, let him answer some of the questions now.
Sure. Well, welcome. Have you been listening into the whole chat?
Yes.
Oh,
I'm so sorry. Welcome to Preeny and her eagle eyes. She managed to spot you.
Mm.
Well,
So-
welcome.
Well, one of the things that interested me is this question about, the actual impact on fish. Are there any real observations of how they respond? I did quite a lot of work on tracking cod and haddock on the seabed and then looked at the effect of sounds on them. They occupy territories. And often if you played back sounds, they moved away from their territories. And that was a real disadvantage to them because they within their territories, they did all their feeding and so on. And I think the same thing happens with their spawning grounds too. If there's too much noise, it drives them away from their spawning grounds. So they don't go to other areas to spawn. So they can't spawn if they're driven away.
Yeah, no, that's absolutely a good point. And I know that I worked quite a lot in Namibia, and there was quite a lot of interest there in the effect on seismic work, particularly on tuna and other commercial fish species, and people desperate for information about this, and it's an area that we desperately need more work in. Yeah.
Yes.
I'm gonna. Is there any other points that we sort of covered, Tony, that you feel that you would like to chip in on? Because we've been chatting pretty freely. I've asked some pretty dumb questions, I think. But
I don't think there's anything else I need to add. I think Art has given very good answers to all your questions.
Okay, and did you hear the section where I said thank you so much? Because I mean, it was just so lovely to have your plenary.
Yes.
I've got to leave. I've got something else I'm about to do. But
no problem. Then I'm going to wrap it up.
But I just want to address, you asked about using our presentation. I don't know if Tony heard that comment, but I have no problems at all, as long as you let us know where you put it. Tony, do you mind if they put up or if they want to do it, put it on YouTube or something?
No, I think it would be fine. I did notice, though, that some of the sounds that we inserted into it weren't actually played back.
That was my ineptness with how to do this thing.
Yeah. I mean, the sounds when the male haddock mounted the female, those sounds weren't played back.
I have no idea.
And they're much faster than the sounds that were presented.
Yeah.
When the cod is on the seabed defending its territory from other males, it just makes these repetitive sounds. But when females appear above them and come down, the males then go up to the females and start dancing with them, and they make much faster sounds, much more interesting sounds. They make hums.
We did have a movie night where sounds could be submitted. So if you'd like to follow up on that and submit a few sounds for that, then we'll put them on the conference site. We had a sound yesterday that was, what was it? Is it a cow or a tiger? Everybody knows, cow or tiger.
Yeah. I've got lots of different fish sounds though, so it's difficult to know which ones to send you. I've got haddock sounds that were recorded in an aquarium and also in the sea. Particularly the sound of spawning haddock in the sea off the coast of Norway, or actually in a fjord within Norway.
Well, yeah, I mean, a couple of examples I think would be really, really useful. And so, yeah, we'll chat about that offline. So I don't want to hold you up if you need to go. I know you've been watching the results very closely.
Yeah, no, I got to say another thing. But thank you very much for giving us the opportunity to present here. It was a lot of, I really enjoyed putting together. Tony, it was a challenge for Tony and I. We've never done that before, but we worked together. We've never tried with sound and stuff. So I'm about to go off and do another one for another meeting. So
yes, I've just been doing some work for another meeting. I've got a whole series of slides about the importance of particle motion reception to fishes and invertebrates. We didn't really say much on that, did we, in this talk?
No, we didn't. We didn't have time.
We did
miss 2 hours.
It's a really important thing that fish and invertebrates are sensitive to the particle motion and not the sound pressure.
Yeah. So I gotta go.
No one ever measures the particle motion, except me and you.
Yeah. Okay, everybody, take care of yourselves. And Tony, stay hi to Sue. Bye.
Yeah. And
stay on for one more question. We do have one, so you're welcome to stay on.
But we'll say bye.
He had to rush, I can tell. So, I've got this question that I'd like to ask you, and it says, myself and colleagues have acoustic recordings from across the Western Indian Ocean that we've collected on multiple reefs over the years. The studies we collected were for humpback whales, but there's lots of reef fish sounds on the recordings. So are they not designed for fish? How could these be useful?
How could what be useful?
The recordings. So I think it's somebody who's got a lot of passive acoustic recordings, database. So they've got in fact, I know it's Jill, so I think she's listening in. So Jill's been working in offshore in Tanzania, or coastal Tanzania, I should say. And she's got absolutely stacks of long-term passive acoustic data collected because Jill's project was looking at whales and dolphin presence, but also she's presented now on dynamite fishing and detecting dynamite fishing. But her question really relates to the use and scientific use of these data in terms of maybe fish behavior, looking at cycles in fish behavior and things like that. How these data best used?
Yes, it's really important to do passive acoustic recording. I've done quite a lot off the coast of Ireland, and I've recorded the sounds made by seals, whales and dolphins, and by different species of fish. And because I've studied these species in the aquarium, I've known which sounds are from which fish. But I also, a couple of years back, I recorded some sounds from squid. You know, some squid. I had a camera underwater as well as a hydrophone. Wow. And a couple of squid suddenly passed by making sounds. And it was really interesting, you know, and I then Googled to see if anyone had ever done any work on squid sounds. And there was just one paper in, about 50 years ago from Australia saying that squid made sounds there, but nobody believed it was true. But I did record these sounds from squid. So I've got a little movie with the squid appearing as the sounds are produced. So it's important, I think, sometimes to use underwater cameras as well as to record the sound, because then you can see some of the animals.
That's a good point.
Yeah, I mean, the movie night that we've had as part of the conference is exactly that, these sort of unusual observations. But I think that in terms of the film work, I think you're absolutely right. And there's a lot of work now, isn't there, on underwater BRUV, so these baited underwater cameras, which, to my knowledge, most don't have hydrophones, but some do have hydrophones attached, and then you would be able to see perhaps what species are there in the area making the noise.
Yes, that's right. And very often some animal sounds are so loud that they spread over a large area, so you don't always see the ones that are making some of the sounds. So snapping shrimps, for example, off the coast of Ireland, you hear snapping shrimps almost continuously. And then you'll hear a seal, and then a fish, and then, then a squid.
The squid I'm very interested in. Yeah, I know that Jill's working in the work in Zanzibar. There's a lot of snapping shrimp in those recordings as well. Having worked there myself, and the overlap between those snapping shrimp and the dolphin echolocation, which was quite a headache, really. And that actually brings me to another question about fish acoustics and perhaps using maybe biodiversity sort of indices, so acoustic indices of biodiversity, without actually knowing which species are present. And we did have Jerome Suhr talking 2 years ago at the conference specifically about that, but obviously with more terrestrial species. But I think there's a lot of room for development for those biodiversity indices for underwater soundscape monitoring, and particularly in areas like we're working in South Africa where we don't know what species are really being vocal.
Yes, it's not just a question of species either. In case of some fish species like the haddock, different individuals make slightly different sounds. So I wrote a paper a few years ago about distinguishing between different male haddock. And the females, as they swim down from midwater to the seabed where all these males occupy territories, they listen to the sounds from different males and they decide which one they want to mate with, and then they go to it. And usually I think they're listening for sounds that come from the bigger, more mature fish, and their sounds are slightly louder and slightly lower in frequency because they're bigger and their swim bladders are bigger. And therefore, when they make the sounds, they're lower in frequency. So the females are able to tell which males they want to mate with, and they then swim down and choose that one, and it comes up and then dances round with them, and they decide they are actually going to spawn together. And they move up through the water, and she releases her eggs into the water, and the male releases his sperm into the water. So they merge in the water and form little larvae that drift away and then grow into adults later on. So it's quite interesting that the females can actually distinguish different males. So when you're listening to sounds, there are differences between individuals as well as differences between species. So that makes things a little bit difficult sometimes to decide which is which when you hear
absolutely. And it's just, I mean, it's just a fascinating body of work. I could actually sit here and chat to you all day. But I know that we're pushed for time because I think we're actually like 20 minutes over. Courtney's going to tell me.
About 30 minutes over.
yeah.
But thank you so much for coming and chatting to us. And I'm hoping, that you were able to listen into the questions, and you now have access to the whole conference platform, so you'll be able to sort of check out and interact. And if there are extra questions that come through, we'll post them your way.
Mm-hmm. Okay. And I'll send you some recordings of fish sounds, actually. To whom should I send them? Which email should I send them to?
bioacoustics@gmail. The email address that you've been contacting, the African Bioacoustics@gmail.com.
Okay.
We've got your email, so we can drop you an email. And
okay, that would be best. If you send me an email, I can reply to it with some fish sounds attached.
That sounds wonderful.
Yeah.
It has been an absolute honor to speak to you, and it's been really fascinating. And the talk, as we said, is just such a useful resource. So we'll post that with your permission, of course. And thank you so much, and have a lovely evening.
Thank you, Tony.
Well, thank you for organising such a good meeting. You did really well, I think. It's fantastic. Yeah.
Thank you. That means a lot.
And there's a need to have one in the UK as well, actually. Now, there is one. The Underwater Sound Forum is having a meeting on underwater sounds at the end of this month, and I prepared my presentation for that.
Great. That's awesome.
Okay.
Great. Will that be online as well?
Will it be
online?
Yes. Oh, yes. Mm-hmm.
Okay, well, maybe we'll be checking that out.
Yeah, definitely. That sounds amazing.
Yeah. Thank you so much.
It's taking a whole day, actually. I think lots of people are going to be speaking about underwater sounds.
Check that out.
Okay, so I'll leave now then and try and find some fish sounds that I can send you.
I'm going to be following up with you, don't you worry.
The problem is I can find lots of them. It's knowing which ones are the best ones to send you.
Yeah. I know that problem. I'm also very aware of how long it takes to put these plenaries together, and the work that goes in. And it's a whole new world that we're in right now with these online conferencing, and it's a massive undertaking. So we're very grateful to you both for doing that.
Okay.
Thanks so much. Okay, we'll leave it there.